think, given the current political structures of those two different governments and countries, that it might be the opposite. And so why is Canada going on? The quick answer is you're going to take a look at the map of Canada, that most of the provinces have actually managed to make good movements on its climate or emission reductions. There are two provinces that have not, and most of that's around energy production in those two provinces. That's there's a quick answer to whoever answered that question. Our next speaker is actually from the United States, so he might have his own editorial on that, or may not. So it's fine. I want to do a call out to Ms. Fiala and her high school students that are in attendance this afternoon. So we really appreciate you guys taking the time and having the interest to come down to this presentation. I'll let you know that I'll implicate David and Katrina that if you have any further questions about this and want them to come into your classroom, they love going into classrooms. I don't mind either, but they're better at it than I am. So, quick one about Georgian Bay Bias Reserve is that a lot of this work that we've been doing has been grounded in the State of the Bay program, which again is about bringing knowledges from different works like charitable foundations, NGOs that are doing great research work, government agencies, universities, bringing that together and distilling that work. And so we have managed to release 2, and as I mentioned, we are interested in releasing a third. But this also takes the support of folks like you. So if you like this kind of work, this kind of orchestration work, then we encourage you to consider a donation amongst all the other great things to donate to, such as you've heard about today, and will the rest of the time. We are going to be hearing from a fellow from the United States who drove a considerable distance to be here. We're really grateful for that. David, or Bo Bunnell, has been a research fishery biologist at the USGS Great Lakes Science Center in Ann Arbor, Michigan for the last 15 years. He's an adjunct associate professor at the University of Michigan, where he teaches fisheries science and management. And I'll commit to you now that if you make your way back over here in the summertime, we'll get you kayaking on Georgian Bay. So thank you for coming all that distance, and again, thank you for coming.
Yeah, thank you very much. It's a beautiful place to be. This is my second time to Perry Sound. The first time was by boat back in 2017 through the CSMI that Paul talked about earlier. And it was, unfortunately, I came here by car during the night last night, so I missed a lot of the beauty, I think, that's around here. But tomorrow, I'm looking forward to seeing that drive home and seeing what it looks like from the watershed perspective, because it was gorgeous coming in from Georgian Bay. So as was mentioned, I'm really a fisheries biologist. So although I'm asked to speak about the lower food web, it's going to be with a bias with respect to how that lower food web supports fish and fisheries. So just so you know that going in, and I have a whole cast of characters, people that I borrow data from to provide some of the slides I'll share with you, especially about long-term trends in Lake Huron. And Todd did a really good job of setting up this talk. And really what you can think about is my talk will be more sort of, as I indicated, about the offshore. So Todd talked about sort of the watershed and how the nutrients in the watershed, the gradient as they're moving into the lake and that interface between the coastal lake and the tributaries. I'm going to essentially extend that out into the open waters of Georgian Bay and also into the open waters of Lake Huron, where we have most of the data in terms of trends. So again, from sort of the broader perspective, the lower food web is really thinking about, as Todd mentioned, phosphorus, which is the limiting nutrient for algae or phytoplankton, which is really the base of the food web. And so that base of the food web, how wide it is, ultimately affects how much fish can be supported at the top. Because as you go from phytoplankton or algae at the bottom up to the invertebrates that feed on that algae and phytoplankton, so things like zooplankton, things like diporia, things like mussels that filter out that phytoplankton or algae. Those in turn support prey fish, things like smelt, things like chub. alewife even, cisco, that feed upon those invertebrates. And then finally we have, of course, the top predators in the Great Lakes, the salmon, the walleye, the trout that feed upon those prey fish. So as that base of algae expands, or as it narrows with respect to how much phosphorus goes into the lake, that ultimately is going to limit how much fish can be supported at the top. And so there's inherently a trade-off between the nutrients that come in and support a lot of fish at the top, like Western Lake Erie, a lot of nutrients coming in, a lot of fisheries, very productive, to places like Lake Superior with the least amount of nutrients coming in, and of course the lowest biomass of fisheries. And so as fishery managers and water quality managers are trying to find that sweet spot where you have high water quality, but also can support the fisheries that are desirable by the stakeholders in the community. So with that, really what I'm going to be talking about is what are the trends in phosphorus, what are the trends in plankton, and how might those in the end relate to fisheries production. And so really it's in 4 parts, and I'll try to get through this hopefully without going over time. So the first is just talking about where we have the most data in terms of trends, and that's the main basin of Lake Huron. Secondly, talking about, and that's in plankton. And in the second part of that talk, bring some data now into Georgian Bay with respect to benthic invertebrates that live on the bottom. Thirdly, we'll talk about more of my own research in 2017, where we had that CSMI, or Coordinated Science and Monitoring Initiative. So again, we'll have another one in 2022, and part of this workshop will be informing the priorities for what happens in 2022. So in part 3, I'll talk a little bit about what we did in 2017, and that was really a broad comparison of Georgian Bay, North Channel, the main basin, as well as Saginaw Bay, and how those differ in terms of productivity and how they may, and the consequences that may have for supporting fish. And that's what section 4 is. So kind of a roadmap for where we're going. And I'm going to start off firstly by just giving some long-term trends in the lower food web. Again, mostly based on, almost entirely based on the main basin, and this is through the EPA, US EPA monitoring program. That program is contracted out to Cornell University of New York. And so this is a talk that was developed by Cornell University. It was presented at the State of Lake Huron just about a month ago or so in Saginaw Bay. So these data, as you can see, are all in the main basin, and they're all offshore. So again, what Todd talked about was sort of within the tributaries and the coastal waters, what the nutrient trends are and how dynamic that is as you go from up in the land out to the coastal. And now we're going to say, well, okay, what is it when you're out in the open lake? And this is the Lake Guardian that goes out in April and in August across all 5 Great Lakes and provides these data. So here's what phosphorus looks like. And so phosphorus dates back to the early 1980s. Again, this is the limiting nutrient for algae, and it has come down, slowly came down between 1982 or 3 and 2002 or so, and then it dropped abruptly between 2002 and 2010 or so. A big reduction in phosphorus that sort of caused a lot of concern because we saw changes not only in terms of phosphorus but all the way up into fisheries, things like alewife. And then we have seen a little bit of an increase and then sort of a stabilization. But in general, there is some interannual variability, but we've seen a long-term decline in phosphorus in the open waters of the Great Lakes, about 33%. The algae and phytoplankton here's some Georgian Bay data. If you look at the bottom in red there, I don't know if I can turn around or not, but so in red you see that so we measure phytoplankton or algae, an index called chlorophyll, which has been mentioned quite a lot. So chlorophyll measured at the surface, and this is actually through satellites. So we can look at, with the satellite, we can look at how green the water is, and relate that to the amount of phosphorus, amount of chlorophyll that we actually measure in the water. And you can see in the main basin, as phosphorus has declined, so too has chlorophyll at the surface. But Georgian Bay actually shows a little bit different story. It shows an increase. And that was surprising to the authors, and I don't have a really good explanation for why The satellite is telling us in Georgian Bay that the water is getting greener and there's more algae out there. But measuring chlorophyll or algae at the surface is just a very small part of the story. Most of the chlorophyll, actually, if you look in orange there, so this trend here, you can see that most of the chlorophyll actually is below the surface. It's, I don't know, between 10 and 20 meters or so. It's the peak or the deep chlorophyll layer. That's something not measured by the satellite. So you have to look at the thermal vertical profile of what that chlorophyll or that algae looks like through the water column. And there are data, again, from the main basin, both from Lake Michigan and Lake Huron, and you can see that deep chlorophyll layer also is declining, coincident with that declining offshore phosphorus. We don't have these data for Georgian Bay. So as the water becomes, has less algae and less phytoplankton in it, this was mentioned earlier, in terms of this Secchi disk depth, or maybe, I don't know if we mentioned this before, but this is a way that we measure water clarity. So how clear is the water? And all of you can probably go out and notice whether the lakes are getting clearer or not. So it's a relatively simple measurement. You see how far down you can see that disk, and dramatic changes on Lake Huron. Again, this is main basin data. Just phenomenal increases. The Secchi disk depth has more than doubled since that 2002 shift in terms of phosphorus. We've seen less phytoplankton, less algae, and now that water is much, much more clear. And I'll sort of revisit this again at the end. And I don't think we really fully understand the implications of this water clarity change just yet, because it's happened so quickly. So let's move up the food web a little bit and talk about the plankton. So these are the zooplankton or the zooplankton. I think Canadians tend to say zooplankton. I'm from southern U.S. where we say zooplankton. And so these are data from EPA, and Rick Barbiero likes to he color codes these animals. And so on your left are the cladocerans. These are species that mostly graze upon the algae and the phytoplankton. So Daphnia, Bosmina. And then there's the copepods, sort of the rest. And so these are typically found in deeper waters, colder living species. And if you're a fish, obviously the larger copepods provide more calories, more energy for you to sustain yourself on, but they're also harder to catch. So especially if you're a small fish, the copepods are there, but they're harder to catch. Whereas the cladocerans are not as able to swim around and move as quickly, but they are more dependent on phytoplankton. The copepods are also eat smaller, so they're omnivores, so they also can be predatory and eat other smaller zooplankton. Big changes, again, sort of the big changes not only in phytoplankton, but also now we're seeing big changes in zooplankton. And so that big reduction is in 2003 again, relative to before 2003. And what's missing is that red. So the red, the red was those cladocerans, those slow-moving, phytoplankton-eating animals that are out there. We've lost a big number of those. And most of what's out there now, except for some years like 2012, where we see big pulses. Again, this is just main basin. This is not Georgian Bay. We don't know the Georgian Bay story as well. But in the main basin, big loss of cladocerans, mostly calanoid copepod dominated. So larger animals that provide more nutrition if the fish is able to capture them. How many people have heard of mysis? All right, that's good. So mysis is a native freshwater shrimp, and these are awesome fish. Love to eat mysis. I mean, you can see these with your eye. These are, between 5 and 10 millimeters, so really big, full of lipids, full of energy for these fish. And one sort of somewhat good story here is that, again, in the main basin, mysis don't appear to have crashed like we see for a lot of the other animals out there. Mysis seem to be relatively stable, and these are animals that not only feed upon phytoplankton or algae, but also on smaller zooplankton, and that changes seasonally. So this is a good news part. And so if we sort of do a report card, we see a lot of declines. The only increase is in water clarity and no change in terms of mysis. All right, so let's go to part 2 then. Let's now not look up in the water column, but let's look at what's on the bottom. And Todd's talked a little bit about cladophora and mussels living in the near shore. And so now let's look a little bit more offshore and look how that benthic invertebrate community has changed. And these are data also, these are done every 5 years. This is a lake-wide survey that's done, I think it became lake-wide in 2012, for the first CSMI year. So you can see the coverage. There's a fair number of holes, I think, in Georgian Bay, sort of as you go offshore. But there's still enough samples in Georgian Bay to get an index, I think, of how that benthic invertebrate community is changing. And these are data provided by Sasha Karatayev and his colleagues at Buffalo State University, who have a contract from EPA to go out and do this work every 5 years through the CSMI process. And just like I've said before, the big take-home message is change. So in 2000, now we're just focusing in on quagga mussels. So how many people know the quagga versus zebra mussel story? That's pretty impressive. So the zebra mussels are really gone from the Great Lakes. It's now a complete quagga mussel story. So they're the same genus, but they've outcompeted. Well, we don't fully understand how they outcompeted zebra mussels, but quagga mussels are, as you can see, they've gone from barely anywhere in 2000 to there's really no depth at which these quagga mussels cannot colonize and exploit. And this is really bad news because those zebra mussels were really restricted to the shallower regions and they needed hard substrate to attach. Quagga mussels have no such limitation. They can survive in cold water, they can survive colonized sand, muddy substrate, so they are really bad news. And you can see that they're everywhere. Just a few notes though. So if you look at Georgian Bay, most then sort of on the western side. And then the other thing to note later is North Channel has very low densities of quagga mussels relative to the rest of Lake Huron. But we've heard, Todd mentioned eastern Georgian Bay, and again, this survey picked up very low densities of mussels. And as Todd mentioned, that's probably a really good thing because if you had higher densities of mussels, you could get more of that benthic cladophora, which can be quite a nuisance. I'll just skip this one for the sake of time. But the other thing that they looked at in terms of if you look at the community as a whole, this is an ordination plot that you don't have to sort of wrap your head completely around, but just look at how the red triangles and the inverted pink triangles overlap. That basically is saying that the community of benthic invertebrates, which would include oligochaetes, chironomids, mussels, and diparia, that community composition is very similar between Georgian Bay and Main Basin. And it's blue up there in North Channel and green in Saginaw Bay, very different communities. So it's the domination of quagga mussels and that stylo, I don't know how to benthic invertebrates, I'm not even trying to say that word. But those two species that sort of dominate how those two why those regions are very similar with respect to their community composition. So there are some data looking just at Georgian Bay. So I guess I was wrong. 2002 was the first year that Georgian Bay was included in this survey. And so you can see that, again, on the left-hand side, you see the scale is only at 20 per square meter, whereas on the right-hand side, the density goes up to 600 per square meter. So as zebra mussels declined from 2002 to 0 in 2017, quagga mussels peaked in 2007 at the sites in Georgian Bay and have since come down and appear to sort of have reached some sort of equilibrium. If we look at the native species, so these would be diparia in your top left. These are just like mysids. These are really important prey for fish to eat. They're just like hot dogs that live out on the bottom. High energy resources for fish. and it's a tremendous loss for fish food that dipraia is gone now, for all intents and purposes. So we can see dipraia is really no longer. Spheridae, these are fingernail clams, also probably been outcompeted by the larger quagga mussels, very low densities of Spheridae now in Georgian Bay, whereas the worms, the oligochaetes, have increased, and chironomids, so these are essentially the larval stage of the midges that you see when the midges hatch, like in the spring, May, June. Their larvae are important aquatic prey for fish. They seem also to be doing well with a fair amount of interannual variability. So to add there, dipraia, that's a really unfortunate loss of dipraia that we've seen not only in Georgian Bay but also in the main basin. And quagga mussels are still increasing in the main basin, it turns out, because they're still marching out into deeper and deeper habitats, whereas they've been decreasing in Georgian Bay, which is a good sign, I think. All right, so the third part then is to look at some of the work we did in CSMI in 2017 and to compare the productivity across the different basins of the lake in one snapshot year where we had a lot of different agencies out there compiling resources to compare spatial differences. The design of the survey was to essentially cite what we call transects associated with rivers, to look for gradients of near shore to offshore productivity that we thought might be able to, we could trace back to the extent to which river inputs are leading to higher productivity in the near shore, and then seeing if we can see any signal of greater productivity in the offshore, if it's at a place where you have high phosphorus loading coming in. Now, we did a similar design in Lake Michigan where there's a whole lot of big rivers that are, dumping in tons and tons of phosphorus. It was tougher on Lake Huron because it's probably a good thing, but you don't have as many rivers that have a lot of phosphorus coming in. So I'm not going to focus so much today on sort of the near shore to offshore because we didn't really see many differences. And when I say near shore, we're in large research vessels that can't go in shallower than about 15 or 18 meters. So any signal from the rivers, we're probably missing because it's already diluted by the time we're sampling it offshore, say the Thessalon River or the Nottawasaga River. That signal from the river probably has already been diluted, at least in our sampling. So we did this monthly between May and August. From the sites you see here, and 3 did include Georgian Bay, three transects in Georgian Bay for a total of 9 sites. And again, these are some of the signals you'll see in the later plots. North Channel, NC, GB for Georgian Bay. We broke up the main basin into northern and southern basins, and then Saginaw Bay, which I don't have as much data for, was SB. So at each site, we collected water for nutrients and chlorophyll A, which is that index for algae. We looked at the thermal profile of temperature and algae. There was a big focus in 2017 on larval fish. This was as I talk about later, we had a hypothesis that these changes in plankton that we're seeing could be having the biggest effect on the survival of larval fish, growth of larval fish, these baby fish. And if those babies are not surviving as well, that could be explaining why we don't see as many larger adult fish, which is a trend that we're definitely seeing within Lake Huron. We sampled the zooplankton with a couple of different mesh sizes, targeting the really, really small zooplankton that I'll talk a little bit about in a minute, as well as the larger zooplankton that I referred to in some of the earlier slides. And then we also sampled adult fish with different trawls. So you can see. So here we are sampling larval fish at the surface, and then we had nets like this that we could fish down to about 40 meters and then work our way up in the water column, because some of the larval fish are deeper in the water column, others like the light better. Okay, so the first thing we looked at is how does, just again, we'll start at the base of the food web, how does the chlorophyll or the level of algae vary across our sites? We had the expectation that North Channel would actually be the most productive based on satellite images of North Channel looking to be a lot more green than Georgian Bay or the main basin. And in fact, that's what we found when we looked at the mean of chlorophyll throughout the water column. But the signal was really only there in June. So in June, North Channel had more chlorophyll than all the other sites. And you can see another sort of common theme to the talk is that Georgian Bay tends to be the lowest in everything. So in this case, it's lower in June and in July than North Channel. So it's sort of sister embayment. And again, one explanation for this, we think, is that there are no quagga mussels in North Channel. So therefore, the quagga mussels are not filtering out that filtering out that phytoplankton or algae like they are in Georgian Bay, even though the densities are not as high in Georgian Bay as in some areas of the main basin. So total zooplankton, this is all zooplankton combined, so the calanoids, the cladocerans, even the rotifers and veligers that I'll show you a little bit about in just a second. If we sum them all up, and we looked at it by month, we didn't see any differences across all our sites. So there appears to be as much zooplankton out there in Georgian Bay as in the rest of Lake Huron. If we had data for Saginaw Bay, I expect Saginaw Bay would have been significantly higher, but we don't have the data for that just yet. So now I'm going to break down the zooplankton into a little bit different categories. We still have the copepods that are shiftier and harder to catch on your left now. We still have now, just to wake you up, now the cladocerans that move to your right. So remember, those are the ones that feed on the algae. That's a picture of a daphnia there. I've added two groups now. The predatory cladocerans, so these are the invasive water fleas. So this is the spiny water flea. This loves to feed upon bosmina, daphnia, and then a lot of these very small zooplankton that I haven't talked about yet. So on your far left, that is a rotifer. In the middle there, it's a nauplii. So this is a nauplii. It's actually a larval, the earliest life stage of these copepods. So this eventually will turn into something like this. And then on the far right is a veliger. This is the larval stage of our friend, not our friend, but one of our main protagonists, the quagga mussel. So quagga mussels release these veligers up into the water column, and they are planktonic. And they hang out, and this is a way that they can effectively spread as well as they've done. And then through time, they're going to build up that shell, and that shell is going to weight them down, and then they settle out on the bottom. But they have this planktonic larval phase that we can pick up in our zooplankton net. So one thing we did was look at ask the question for a given month, are these different categories, are they varying? Since we didn't see any differences for total zooplankton, now let's break it out and see if we're seeing differences across the regions. And in May, we did find that herbivorous cladocerans and the group of small zooplankton, in fact, did vary across the regions. And if we do that for the other months, again, we found some differences, but not as many as we were expecting. We expected to see more differences in zooplankton across the regions than what we saw. Okay, so this is a little bit busy, but again, so this is now just showing you when the differences occurred, who differed and why. So these are kind of like the other slides I showed you. So on the x-axis we have North Channel, the two North and South Main Basin, and then Georgian Bay is always on your right. So the take-home message is that when we did find differences across our regions, again, it was Georgian Bay that tended to be the least productive. So Georgian Bay had fewer of our small rotifers and nauplii and veligers, for example, than all the other regions. Well, in fact, in that case, just the different letters indicate differences and significant differences. So different than southern Main Basin. Here's a really obvious one. So Daphnia, bosmina, those herbivorous cladocerans, almost found none in May in Georgian Bay, so significantly lower than all the other basins. The same thing again here. So again, herbivorous cladocerans, daphnids, bosmina, very low in Georgian Bay relative to the other parts of the lake in 2017. So what about those veligers, that larval stage of our friend, or our protagonist, the quagga mussels? No differences across the lakes, or across the regions. And to us, this was really surprising because we were expecting to see no veligers up in North Channel because we found very few adults. So what this is telling us is there's a consistent propagule pressure of these invasive mussels up in places like North Channel, as well as in Georgian Bay, where their densities were really low. But so there's something else that's limiting their survivability, and Todd showed you this too, it's calcium. So they need calcium in order to build their shells, and what we found on your left there is that the calcium was always the lowest in North Channel. The other place that it was relatively low was in Georgian Bay. So in Georgian Bay, calcium is almost always measured at less than 25 milligrams per liter. And we unfortunately do not, to my knowledge, we don't know what the lower limit is for quagga mussel veligers to survive or for adults to occur. For zebra mussels, there's evidence that it's 25. So quagga mussels, another potential competitive advantage is that they apparently can survive at calcium levels lower than 25, because in general that's what you're seeing in offshore Georgian Bay. All right, so then as we wrap up here, let's talk about how all of this relates to fish. So how do these differences in productivity, or how does the long-term decline in productivity affect the ability of these different regions of Lake Huron to produce or support more fish and fisheries? So we did this really two different studies. The first, we looked at a species called rainbow smelt, and we asked the question, does rainbow smelt in 2017? So our hypothesis was that in regions where you had more plankton or more zooplankton, that you would find rainbow smelt in higher energetic condition, because essentially there's more food for those smelt to eat, if that makes sense. So there's a picture of rainbow smelt, and I want to credit Chiandet, who was a graduate student of mine at the University of Michigan, for doing this work, and that is the biggest smelt you'll have probably ever seen, right? No, it's not a smelt, though. That's a lake whitefish with a nasty sea lamprey wound on it, in fact. But Chiang was really excited to see something bigger than a smelt, so we took a picture. So smelt, why do we pick smelt? It's not a native fish. Oh, well, we picked smelt because it was ubiquitous. It was a species that we found consistently across all three basins and in Saginaw Bay. The other cool thing about smelt, I don't know about cool, but the other thing about smelt that was cool for our study is that it also occurs in Lake Erie. So we also looked at the smelt energetic condition in Western Lake Erie to give us the widest gradient of productivity, because Western Lake Erie is a place where there's the most phosphorus, the most plankton. So if we were going to see a difference, we expected to see it there. All right, so the take-home message, I'm going to break this down by month. And so what you're seeing in April and May in the top and July below it is, and so on the x-axis is the length of the size of the smelt, on the y-axis is its energy density. Energy density, what does that mean? That's essentially how many calories is packed in that smelt. So, we buy a Twix bar, we're eating 200 calories, right? So now we're saying in a smelt that is relevant to, for a lake trout, let's say, when a lake trout eats a smelt, how many calories is it getting? And does the number of calories vary across these different regions? And what we're showing you is that it doesn't. That a smelt in North Channel has the same number of calories as a smelt in Georgian Bay. Even though the productivity in North Channel was about twice as high as it is in Georgian Bay. So that was surprising. When we add data from Western Lake Erie, however, that's when we did see differences. So the smelt in western Lake Erie had about 30 or 35% more calories. We say joules, kilojoules, but calories I think is easier to understand, than all the smelt in Lake Huron. So it does suggest that the smelt in Lake Huron are not as calorie-packed or energy-rich as they could be if the lake were more productive. The other thing we did is we looked at our study in green there relative to other studies done going back into the 1970s throughout the Great Lakes. And what you can see is that the energy density we measured for smelt in Lake Huron in 2017 was at least 30% lower than has ever really been measured in other studies, be they in Lake Ontario or Lake Michigan or Superior or Huron. Again, suggestion that the smelt do appear to be limited by the productivity of the lake and aren't able to pack as many calories in there as they used to. And that includes, this is another Lake Huron study from the 90s. So the second part, running out of, as we get closer to our time here, is this idea of what's called the larval bottleneck. So I referred to this earlier as this, when baby fish, so if we just think of a simple life history model here. So we have an adult, this is a picture of an alewife. She releases eggs. Those eggs hatch out into a yolk sac larvae. And those yolk sac baby fish, have part of that egg that supplies their nutrients for the first several days of their life. So it essentially allows them to transition into this very different environment, gives them a few days before they have to transition and feed on their own and find the right size food and be able to capture the right size. So find the right size food and then be able to capture that food. How well are they able to do that? And because the zooplankton community has changed so much and we've lost those easier to catch cladocerans, we hypothesized that this could be that these larvae are growing more slowly, are perhaps starving more frequently, and their survival is lower, so therefore you have fewer juvenile fish that survive to be adults and can allow this life cycle to continue. So we call this, we're looking for sort of a bottleneck in their survival. And so it's kind of cool, larval fish, just like when you go out and, if you're looking at wildflowers, there's a group of spring wildflowers, summer wildflowers, and then some that even bloom in the fall. So the fish community is also that diverse. We have some that, like lake whitefish, that spawn early. Then we come up with burbot, and rainbow smelt tend to be most abundant in the period between May and July that we were sampling. So again, we sort of fell back on using rainbow smelt for our study because they were the most ubiquitous. And so what we looked at was their growth rate. So people with much steadier hands than me were able to, so this is a larval smelt, were able to dissect the ear stone or the otolith out of these baby fish. And just like a tree, there are rings that are laid upon this otolith. So there's essentially in a larval fish, there's a time during the day where they're feeding, and then at night they're not feeding, and that sort of diel pattern leads to these daily rings that get laid down. I don't know if you can see it, but you can't see very well from here. But under a microscope, you can count how many rings you see. And for that particular one, that's about a 36-day-old fish. And so we know how long it was, and we know its age, and we can figure out how many millimeters it's grown per day. And you can see these are the densities of smelt at our different sites in 2017. So a lot of smelt up in North Channel and in some of northern Georgian Bay, near shore Perry Sound. Not as many smelt in Main Basin. And then, of course, there's this relationship in general. If you look at age versus length, the older that you are, on average, the longer you tend to be. But what we're interested in is explaining this variation. So some, 30-day-old fish are relatively, 27 millimeters. Others are like less than 20 millimeters. So what is it about the environment that allows some to grow larger, others to not grow as fast? And so some of the hypotheses we looked at were water temperature, we looked at how much algae there is as sort of an index of productivity, how much zooplankton is there, so how much food is there for them to eat. But remember, we only can do that on the day that we sample the fish. We don't really know how much zooplankton there was, their whole life. We just know it on that day they died, essentially. And then we also looked at sort of, is there a competition factor? So how many other smelt there are for them to try to find and compete with for food. So we used a statistical model for that. And what we basically found is no relationship with the amount of food on the day they died. So this was sort of just very disappointing to us. We couldn't find a signal for more food means faster growth rate. And again, part of one explanation could be that because we only sampled it on the day they died and not the first 30 or so days of their life. But we did find a pattern as we would have expected. So in places where there were more larval smelt, they tended to grow slower. So this is consistent with some resource limitation evidence, but not the full pattern that we expected to see. So in conclusion, sort of high-level findings from what I talked about. In general, I've described a lot of changes. So the nutrients and biota in the lower part of the food web are trending downward pretty consistently in the main basin, and I think we're fairly confident that similar trends are occurring in Georgian Bay. When we looked at, in our 2017 CSMI year, Georgian Bay consistently came out as the lowest among the different places that we studied. We think that calcium probably is limiting quagga mussels from being even more abundant in Georgian Bay, which is probably a good thing. And we are seeing increases in things like the worms, the oligochaetes. And when we look for evidence that reductions in the lower food web are having an effect on fish, it's sort of a mixed bag. We do think there's evidence that is the case for rainbow smelt for adults because they aren't attaining the higher levels of calories that they used to be attaining. But we're not finding the evidence so far of sort of a bottleneck for survival in that very early larval life history stage as we had hypothesized. So a couple of things to think about maybe for breakouts, what things we might consider for 2022. I alluded to the fact earlier that I don't think we have a really good idea of what's going on, like the effects of the changing water clarity. We have a doubling of water clarity throughout most of Lake Huron. And one thing that we haven't been thinking about that we're going to begin to think about in Lake Michigan in its CSMI year of 2020 is the understudied role of ultraviolet radiation. So we put on, to protect our skin from damage and from cancer, we wear sunscreen, right? Especially people as pale as myself. And so fish don't have sunscreen, but how does it come across? So you can see the image on the right. That is a native fish, and you can see that's a bloater. You can see all the melanophores on it that can protect it from ultraviolet radiation. The smelt on your left definitely does not have any sort of pigment that can protect it from ultraviolet radiation. So as the lake is more clear, ultraviolet radiation is penetrating deeper and deeper. And what we don't know is whether or not larval fish are adjusting their depth in the water column to protect themselves from that ultraviolet radiation, or alternatively, if this is a source of mortality that we haven't been tracking. Another thing that came up in our State of Lake Huron conference was lake whitefish recruitment. And this was alluded to earlier, and I'm sure Arun is going to talk about it too. So large declines throughout Lake Huron, including in Georgian Bay here in black, as you see, the yields of lake whitefish have been declining. And this has consequences for First Nations and tribes in the U.S. as well, because this is something they certainly rely upon heavily. So there's a couple different hypotheses. One, we think that there's still that larval bottleneck that I talked about, looked for evidence for smelt. Maybe this could also be important for lake whitefish. In general, we expect that there's the least amount of plankton available in the spring, just given the seasonality and zooplankton. But we think that spring may be affected even more because of mussels out there. And so the biggest change that we've seen with respect to phytoplankton is in the spring, because that's when mussels are filtering out the most phytoplankton from the water column. And so when we look at the relative abundance of zooplankton in May versus June and July, and I'm just showing you now the nearshore sites in Georgian Bay, again, evidence that there's not much food out there. In the 18-meter site, our shallowest site, that lake whitefish could be feeding upon. So we don't know if they're growing more slowly or potentially even starving. This is a potential hypothesis. And I would say we also aren't even sampling where these lake whitefish larvae are. We're out in 40, 50 feet of water. These lake whitefish tend to be found on beaches, so in really, really shallow water, places where you can seine, and we don't have any sort of data on food or zooplankton. The other one is that we think that, sort of related to climate change, we know that there's strong evidence that lake whitefish tend to recruit better, produce stronger year classes in years in which there's more ice cover. And David provided some really cool, not only an inspirational talk with respect to climate change, showing you not only at the global level but the local level some of the things that have been changing, and that's definitely true. And when I looked, however, at just the Lake Huron data, and I didn't spend a lot of time with this, and I'm not even sure that there are data for Georgian Bay in particular, but there is no trend, surprisingly, when you look at maximum ice cover in Lake Huron. And I think there would have been a trend had we sort of stopped here, but we've had a couple of years with much higher ice cover that has caused this to no longer be a trend up through 2019. So there are other measures of ice cover than just percent maximum ice cover that I think we would see evidence of climate change, thickness of ice, time of ice formation, but from the data that was available to me through, over the weekend, I couldn't really pick something like that out. But I think it's something to think about as another potential driver to explore what role is climate change having on lake whitefish. And with that, I just want to acknowledge our vessel crew and the other vessels that spent so much time in our CSMI year sampling the different regions of Lake Huron. Thanks.
I'm thinking back to on your table, you'll see, and you've got one out already, the food web. Was needed for that presentation. So, thanks. Do we have, I think we have time for one question if there's anybody with a burning question right now that would like to come forward. No? All right. So what I'm going to ask you to do is I'm going to ask you to stand up. Thanks. Everybody stand up really quickly. Do a turn. Don't get dizzy and fall down on me. All right. I'm thinking about with the climate change work and the invasive species work and some of this work that Bo was demonstrating around how much of it is driven by invasive species and the cost to try to rectify these kinds of ecosystems, let alone understand the cost to understand where they're shifting and where they're going to land. Is so much more once it's been damaged than if we had sort of just hit a pause and stopped ourselves way in advance. And I was reflecting during that talk, just like, when are we going to learn that lesson of like, whoa, like stop for a bit. Like, what does this mean? What are the ramifications? Maybe we shouldn't be doing this just now because, man, when we take a look at climate and I take a look at some of the youth in the rooms, like. We can talk about financial deficits, but man, we are creating some really wicked ecological deficits for our later generations. So on that really sobering thought, thanks again, Bo, for your presentation. We're going to shift now, and I'm just going to move this computer over so we can do the shift.
He wants you to leave it there.
Oh, okay.
I'll put it back.
Yeah, we'll get it back. You can have a seat at any time. I love the fact that you're still standing, though. One of our previous directors of the Bias Reserve was also he did strategic planning for us at the Bias Reserve, and he would take time after lunch to go and kind of just lie down, and he would put us to work. He's still standing right now, which is just fine. Aisha is coming from another area where if we had done things better from the get-go, then we wouldn't have had to spend the years of work. So she's coming up from Severn Sound, which was previously an area of concern. And she's been working down there for a number of years, has been working with GBBR and our State of the Bay report and many other areas, providing incredible presentations and incredible resources for us to share with you. And the other kind of cool thing is that if you were around about a week ago, she might have been suspended and coming down from trapezes from up here. Trapezes? No, not trapezes. Basically, she's an aerial circus performer, and she was here a week ago performing, which is kind of cool. I would ask for a repeat performance, but I'm not entirely sure that can happen. So anyway, round of applause for Aisha. Thanks for being here.
Looks much different in here than it did that night.
Back up.
Oh, yeah.
Greg didn't listen to you. There we go. Getting there, getting there.
Okay. Good afternoon, everybody. Thanks for sticking around for the whole day. This is a lot of information to digest, and we appreciate you guys putting in the full day. So I'm from the Severn Sound Environmental Association. I work as a water scientist there. And thank you to Bo for setting up sort of what I'm going to talk about. So we were talking about the offshore communities and trends happening there, and I'm going to shift the focus and look at more at sort of a detailed nearshore area, so focusing on Severn Sound. So Severn Sound, as Greg mentioned, was an area of concern on the Great Lakes, listed back in the late 1980s, primarily due to concerns over nutrient enrichment, particularly due to phosphorus, and also the algae proliferation that resulted from that. So delisting was achieved in 2003, so after a remedial action plan process was followed. But a condition of that delisting was that we continue doing the same type of monitoring to make sure that we don't slip back to those previous conditions. And so the Severn Sound Environmental Association was sort of born out of that process, and we continue to do the monitoring and stewardship work to this day. So since data was collected back in the early 1970s, initially by the Ministry of Environment, Severn Sound has seen a lot of dramatic changes. So partly due to the reductions in phosphorus loading, so things like upgrades to wastewater treatment plants and private septic systems, stormwater management improvements in places that there was no stormwater management, agricultural best practices, tree planting, all that good stuff, really made an impact in the amount of phosphorus coming into the system. But as we've heard, the zebra mussel story and quagga mussels impacted Severn Sound as well. So that was about mid-1990s that mussels came into Severn Sound. And although the densities still aren't as high as what you see in some of the lower Great Lakes, it's still enough to create quite an ecological change. And so there was really quite a big shift, sort of pre versus post mussel invasion. And so this talk is going to focus sort of on the lower food web post zebra mussels to sort of get a bit more into some of the more recent changes that have happened since the mussels came in. And so looking at this data set, we can look at some different questions, such as how have algae and zooplankton communities changed over time? How are the shifts in algae community composition related to ecological changes, such as the mussel invasion timing and phosphorus? What role does water temperature play in terms of algae biomass? Bo alluded to some of the changes in the timing of food availability for prey fish, and in this case, we can also look at food availability being algae for the zooplankton populations. So how have the timing of those peaks changed? And then also, have there been shifts in the type of zooplankton groups that are dominating? So just to give you a little bit of background on the type of information that we collect. So this is Severn Sound. We're down sort of in the Midland general area. So we collect 14 different stations across Severn Sound. Every 2 weeks we're out there. And these stations have been sampled, some of them since the late 1960s, some of them a bit more recent. So we have data from anywhere from 16 to 48 years. Quite an extensive data set. So in terms of methods, we're looking at biweekly sampling, as I said, through that ice-free period, so from May to October. Zooplankton was added to our roster a little bit later, so more the late 1970s. We collect vertical profiles of physical data, so temperature, oxygen, conductivity, also looking at water clarity. You heard about the Secchi depth. We collect water for nutrients, so looking at sort of the sunlit portion of the water column, analyze for nutrients, and that's also the portion of water that we sample for algae community composition. So those algae samples are, depending on the site, are sometimes counted as the individual dates, or sometimes those are pooled and counted as one annual sample. And then finally, we collect our zooplankton for community composition, and that's throughout the entire water column using an 80 micron mesh net. So kind of somewhere in between the really small critters to the larger ones, we're kind of capturing somewhere in between there. So I just want to set the stage for some of the changes in the lower food web by describing some of the broader ecosystem changes that we've seen in Severn Sound. So looking at the top graph on the left, total phosphorus. So all these graphs are just showing changes through time at some of our long-term sites. And so you can see the declines in total phosphorus. The really drastic one is in lower Penetanguishene Harbor, which was sort of the most eutrophic of all of our sites. But all of the other locations also saw significant declines in total phosphorus. We also had the zebra mussels, so we have villager density on the top right there. So you can see that it was about 1994 that we started seeing villagers show up in our zooplankton counts. And these density data can be a little bit hard to interpret because it's kind of like sampling bacteria. They can be quite variable from year to year. But definitely we know that they were present starting then and have not really let up since then. The lower graph is showing phytoplankton volume, so this is just a total measure, so not breaking out based on the different groups. But essentially we're showing the same sort of trend where there have been significant declines in the amount of algae in Severn Sound. And you can sort of see if you look around 1995, right here, there has definitely been quite a substantial drop in the amount of total algae. And then the last graph at our deeper long-term stations where we can look at water quality. The shallower ones, the disc is actually on the bottom, so they don't count. But the deeper stations, we saw this shift that happened right when the zebra mussel colonization happened. So it was a very large increase in the water clarity. And then since that time, water clarity has actually been declining overall. And then overlaid on top of all of this, we've been hearing a lot about climate change, and so water temperature in Severn Sound sort of gives you a kind of a local context of what changes have been happening. So out of all of our long-term sites, the mean annual surface temperature, so this is just 1 meter below surface, has increased, and over this 50-year period by about 2.5 degrees. And we actually found that the fall water temperature rate of increase was double this. So the late, sort of late summer, early fall temperatures are actually increasing quite a bit more substantially, which has implications for things like blue-green algae blooms. So to dive into some of the variability in the community composition of the algae community in this post-zebra mussel world, and I'll just draw your particular attention to the kind of the light blue, I don't know if you can tell on that screen, the blue-green algae and sort of how that's been fluctuating at some of the different sites. So we'll just kind of go through and bear with me. Some patterns will start to become evident. So in our lower Penetang Harbor site, we have a decrease overall since the zebra mussels came in the total amount of algae. And if we look at the algae community sort of on a percent composition basis, the amount of blue-green algae has actually been increasing as a relative abundance. And also a decrease in the amount of diatoms. So diatoms are an important food source for zooplankton, and also can be indicators of eutrophication. So moving up Penetang Harbor, again we have an increase in the percent contribution of the cyanobacteria, the blue-green algae. Also an increase in chrysophytes, which is a golden algae, and some of those species within the golden algae can contribute to like poor taste in drinking water. So something that we watch out for. And again, that decrease in diatom contribution. Moving up, again, we still see that increasing in blue-green algae and the golden algae, and a decrease in diatoms. In the outer part of Penetang Harbor, it's a little bit different, a lot more flushing from the open waters of Georgian Bay. But the change that we saw there was still the decrease in the amount of diatoms. Moving over a little bit more, again, quite an open site. We kind of use this as one of our reference type sites. And saw an increase in the golden algae and decrease in diatoms. Moving up into the Honey Harbor area, so this area is quite a bit different. We're getting up into the Canadian Shield type rock, so more similar to up in this area, and a lot more restricted mixing. So up here we actually saw an increase in the total amount of algae, and also the increased contribution from blue-green algae. Same sort of story for further up in North Bay. So increase in the total amount of algae, increase in goldens, and decreasing diatoms. You can start to hear the pattern here. South Bay, we definitely see an increase in the total and the contribution from blue-greens. Coming back down into Midland Bay, a little bit different. We didn't see the diatom effect so much, but still an increase in the golden algae. There was actually no significant changes in this part of Midland Bay, which is interesting. Moving over into Hog Bay, similar depth, so increase in the total amount of algae, increase in goldens and green algae as well. Moving further into Hog Bay, we have an increase in that total amount, and the goldens, decreases in diatoms. Sturgeon Bay is a little bit different. It's quite shallow and quite a lot of plants in there, so the algae community tends to fluctuate a fair bit. And we do see in this area that the dominance of blue-green algae can be quite substantial, but it didn't actually change over time. And then coming up into Port Severn, which is actually the mouth of the Trent Severn waterway. So the Port Severn Dam discharges out into this area. So there's quite a large amount of flow, and we don't see as high algae biovolume over in this area. But there was still an increase in the total amount of algae. So when we go down and actually look at the species contribution of some of these groups and sort of what's driving the changes in the fluctuations of some of these groups, we can see, so in particular the blue-green algae, microcystis, which is one of the main toxin producers that gets talked about, In that pre-mussel period, there really wasn't much in the way of microcystis around. And then post-zebra mussels, it's still not in really high abundances, but the microcystis is becoming much more detectable, and definitely something to watch for. And this is well documented in other systems as well. There's some evidence that zebra mussels somehow selectively filter out these blue-green algae. They don't taste very good, apparently. We have other types of blue-green algae that used to be a lot more abundant and then have dropped out post-zebra mussel. So it's sort of the same type of story with some of the other groups, particular species that were a lot more abundant sort of pre-mussel days. And as the nutrients sort of gets filtered out, those algae are dropping out. So a similar story with some of the diatoms, which can be indicators of high nutrient conditions. And so it's kind of showing the story of Severn Sound becoming more of a mesotrophic, and in some cases almost oligotrophic system, definitely moving into that lower nutrient state. Looking at some of the seasonal changes in algae, unfortunately we don't have seasonal data for all of our sites. But we do for our open water, open Severn Sound site going back to 2005, so we can kind of have a rough look at how the changes in the peaks in algae are changing over the years. So we're definitely seeing more cyanobacteria blooming sort of later in the season, which is sort of consistent with our higher temperatures that we're starting to see, sort of into the September-October period. And what we've noticed with this data is we're not really seeing sort of an early spring algae bloom that would feed the zooplankton population. So it's really looking at so more of a midsummer bloom, and it's possible that this mismatch of timing could be translating to a lack of food for juvenile zooplankton. And what we're actually seeing in the zooplankton community is that there's been quite a decline over the years. So starting up in sort of that lower Penistang Bay area, where we had very large densities, but they've since really dropped out. It's really been quite widespread over all of Severn Sound, especially since the zebra mussels came in. And those declines have occurred across all of the zooplankton groups. So that was great that Bo kind of set up these different groups and mentioned that the Daphniid tended to drop out in the open water sites, but in Severn Sound we're really seeing that decline sort of across all groups. I'm going to skip over this one in the interest of time. So just to summarize, that we have seen a very clear regime shift in Severn Sound, and this has been marked by dramatic increases in water clarity, reductions in phosphorus, and reduced phosphorus loading from watershed sources. Long-term monitoring of the lower food web has indicated that this shift impacted both the algae community and the zooplankton community. Algae biomass has dramatically decreased since the 1970s. But since that mussel invasion occurred, the biomass has held steady in a lot of areas and increased in others, despite no increase in the amount of phosphorus. So there's potential influence from increasing water temperatures, growing season, ice cover, all those kind of factors that we need to look a bit more closely at. Shifts in the community composition varies, but generally includes seasonal dominance of different algae groups, so mainly the golden algae, dinoflagellates, and blue-green algae. And these often are made up of nuisance or potentially toxic species. And then finally, zooplankton densities have also dramatically decreased across all major taxonomic groups. And there's a potential influence of sort of a mismatch of the spring bloom timing. So I'll just wrap up by acknowledging the support that we get from our municipal members. And I'd like to acknowledge the mayor of Georgian Bay Township, Peter Kutsier, just at the back there. We get a lot of support from Georgian Bay Township. Also the Ministry of Environment, Conservation and Parks for all our water quality analysis and use of their historic data. Of course, our wonderful staff. If we have time, take any questions.
Mr. Will.
Probably the least scientific person here, so I'll ask stupid questions.
No stupid questions.
Is there an optimum level of algae or just some algae? Like if you were to describe a perfect water quality based on algae to support all the rest of the levels, what would that look like? Like how do we know we're getting rid of the blue-green and we want more of the yellow or the orange?
Yeah, that is a good question. I think sort of not having an overabundance of any one particular group. So when we get a blue-green algae bloom, for example, which is not necessarily an unnatural thing, it becomes sort of a human health hazard when they persist and they're very frequent. But if you look at lakes, way up north that don't have any human influence, they still get blue-green algae blooms, and more so with increasing water temperatures. So I think it sort of depends on the system that you're looking at and the human uses, right? There's sort of that factor that, people don't like the look of a lot of algae, and so even though it might be natural, like this year we saw a lot of filamentous algae growing in the water column in Gloucester Pool, and it's not toxic, it doesn't cause an issue, but people don't like the look of it, so it becomes a management problem. So I think that's the discussion too, is like, is it an ecological problem or a management problem? So sorry, it's kind of a hard question to answer.
Sure. Anybody else? All right. Oh, yes.
This might actually be a question for Bo, but is it possible that larval fish are consuming microplastics?
I would take a shot in the dark and say yes, because I'm pretty sure I've heard research where plastic content has been found in stomach analysis. So, and I know we've seen them like in our zooplankton samples. So it's definitely of the size that fish could be eating. And I don't know if we want to, anyone out there who has any of that data?
We have some confirming nods coming from the back.
I don't know that I've seen it in Great Lakes larval fish, but definitely is theoretically possible, probably likely.
And probably the closer you get to an urban center, fish that might be in Perry Sound where there's a wastewater discharge, more likely versus out in the open waters.
Yeah, Aisha, thanks so much. All right, so I think for me, from more a management planning perspective, I guess, is when I look at the work of Severn Sound and the years of detailed monitoring and research that has gone on with the rich data set that exists there. I wish we had it, for the Prairie Sound, the Shawniga area around Shawniga Island, and again up in Magnetawan, just that ability to describe a north-south trend for eastern Georgian Bay. Maybe I'm just getting selfish for UNESCO Biosphere and research excellence, but I think it would be fantastic to be able to make those comparisons, and certainly having Severn Sound with that rich database is an incredible resource for Georgian Bay. So, again, it's back over to you. This is your turn. We have provided you, again, with a new set of questions. So these are different questions related to this topic. And I would say that if you get into the questions and you don't like the questions we're asking, change them. This is your opportunity to provide feedback into the CSMI. We're simply kind of providing some kind of questions that might help to guide the discussion. Behind me here, Katrina has pulled up sort of, again, that quick synopsis. If we were going to put a few recommendations, 3 to 5, I think it might be on that list, 4, pardon me, forward in terms of what would that research monitoring, those monitoring priorities be. Here are some recommendations that seem to be coming forward, so you can reflect on those as well and get back. I encourage you to do a couple of things. One is that if you're tables that become fewer in numbers, consider amalgamating the tables, that is, and as well, if you have an opportunity, feel free to just pick yourself up and move yourself to a different table so you might engage in different conversations with other people. So again, we'll be circulating around. If you have any questions, we'll find the expertise that can hopefully provide you with an answer. And this will go on for about 20 to 25 minutes or so. All right, thanks everybody.